The majority of drugs which act on enzymes act as inhibitors, and most of these are competitive. They compete for binding with the enzyme substrate in the active site. Some inhibitors are non-competitive, which means they are binding away from the substrate's domain and competing for cofactor or coenzyme binding, or causing an allosteric conformational change in the protein structure that stops the substrate from interacting. Other inhibitors are irreversible, which means they're covalently bound to the enzyme, and this causes a permanent inactivation of the catalytic function. Drugs can also act on receptors in the cells. Receptors are proteins that are either inside the cell or in the surface of it, which receive a chemical signal. The binding of the active compound on drugs results in a cellular effect, which could be altering gene transcription, translation, or changing the cell morphology. For example, drugs that work on the brain interfere with the way neurons send, receive, and process signals via neurotransmitters. Some drugs, called prodrugs, are administered in an inactive form, which is metabolized into an active form. They are expected to enter a metabolic pathway for the resulting active metabolites to act with the desired therapeutic effects. Most drugs must pass through the liver, which is the primary site for drug metabolism. Once in the liver, enzymes convert prodrugs to active metabolites or convert active drugs into inactive forms. Drugs are complex and act in many different ways. They're meant to cause a physical change in the body of the user, which can treat from inflammation to cancer. Talking about cancer, one example of an active component is the writer enzyme, which catalyzes the addition of chemical groups into either the histone tails or the DNA itself, creating epigenetic marks. The histone deacetylases inhibitors correct the imbalance in the equilibrium of histone acetylation that is associated with tumorigenesis and cancer progression. Verinostat was the first inhibitor of this type to receive regulatory approval. This is just an example on how inhibiting enzymes can stop specific reactions from happening that could have been modified after falling sick. This not only stops multiple symptoms, but generates a biochemical and structural change in the body and even our genetic material, as we have previously seen. That was a brief introduction to the mechanisms drugs use and how they change our body. Well, thanks for listening.